welcome everybody. Thanks for joining us, and thank you to the CNPS chapter from San Francisco for inviting me, and especially thanks to Susan, who's being perfect host, supporting me as a speaker for this presentation. And I'm particularly excited because I lived most of my life in San Francisco. I know that people attending this talk are not just from San Francisco, but I'm always very excited when local chapters invite me to give a talk, as I do love my city very much. I'm a professor at Berkeley, but I'm also the forest disease expert for the entire state of California for the UC system. As you know, the UC system is has the federal responsibility to take care not just of agricultural crops, but also of natural resources. And I'm the one person that's supposed to do it for the entire state, which of course we know it's ludicrous, but at least I do what I can. And today I'm going to talk about something that unfortunately it's particularly concerning. And really, I and others have realized that this is happening just quite recently. But at the end of this talk, I hope I will have been able to have convinced you that this is going to be a major driver of change for California, more than sudden of death, more than pine pitch canker. I think what we're seeing here is going to be what is going to reshape our forests, because let's be clear, our forests are going to be reshaped. And we can just sit back and watch, or we can be a little bit more actively involved to the extent that we can and that it's feasible. And I think the knowledge that's coming out of this kind of research is really going to help us to make some important decisions that maybe at the end of the talk we'll go over. Some of these decisions are not necessarily popular, but I think from a scientific standpoint, at some point, we may want to make some decisions that may not be necessarily popular with everybody. And before starting to talk, so yes, I'm based at Berkeley, but again, I work throughout the state. I just wanted to thank the people in my lab that have started this research. This started as a relatively small project that was funded by the East Bay Regional Parks. And 5 years later, here we are. Most of the lab works on this project because this turned out to be a very interesting, fascinating, and very important topic. So Ines Marquez was one of my PhD students from Portugal, and Tina Popenak and Doug Schmidt, some of you may know them. They are the managers of my lab at UC Berkeley. And so in the title, I use some big words, and I really have to explain to you what the words mean, because there's something very, very unique about the way we did these 5 years of research have been done in a very unique way. I've never done anything like this before. So I'm going to talk about novel tree mortality. So this means that, I mean, trees have always died. So why am I calling this novel? Because in the way it's presented itself, it really looks like it was previously unknown. It was unreported, and it's of unknown origin. So it's definitely not your garden variety mortality. It's something really different that's happening. It's at a large scale. So it affects all of Northern California. The locations, and we show you some maps, span in the distance of hundreds of miles. But locally, the effect of these new diseases are very different. So we can't really generalize. Still, the numbers are quite shocking, but again, it could be very different in one side from the other one. So we're not talking about mass mortality of all the trees like it happens with bark beetle outbreaks, or like it happens with sudden of death outbreaks. But nonetheless, the effect is more subtle, but it can be more significant in the long term. And the major players that I will talk about, I call them latent pathogens, and it's not just me. This is a term that emerged in the 80s, and then it reaffirmed in the 90s. These are organisms. Why we call them latent pathogens? We call them latent pathogens not because we can't see them, because that could be an interpretation of the term, but we call them latent pathogen because most of their life, they live it inside the plant as endophytic organisms. And some of them may be beneficial. And we mycologists are kind of lucky because we actually know that there's two family of fungi, actually two orders of fungi, the Dothidiales and the Diaporthales, that contain a very large number of these latent pathogens. So these latent pathogens originally are endophytic. They're part of the microbiome of the plant. They're a very small part of the microbiome. So today I'm not generalizing. about the entire endophytic community. I'm just talking about very specific players. And then at some point, the trigger to become, and they can be beneficial or neutral. And at some point, the trigger to become pathogens, and they kill the plant. And then unfortunately, they actually sporulate, they produce spores that they start reproducing on the dead plant that they killed, or on the dead branches of the plant they killed. And so they move from being pathogenic to being saprobes. And then those spores will infect younger trees, and then the endophytic stage starts again. And what triggers these organisms to shift from being endophytic to being pathogenic? Which is a huge shift. And I'll show you one slide about what it means to be an endophyte. Well, we know that they're triggered by not necessarily climate change. I should have put environmental change. And a lot of it is climate driven. And of course, higher temperatures and water stress are major players. But there can be other environmental stressors. It can be frost. It can even be mechanical damage that unseals the sealed tree. You know, a tree is a sealed system with a very, normally in Mediterranean climates, with very high water pressure. So when you have mechanical damage, you unseal the system, and that actually can trigger these endophytes to become pathogens, at least locally, where the lack of seal allows for oxygen to go in. But even defoliation, for instance, that's caused by a third party, can activate these endophytes to become pathogens. So another term that I use when I teach my class is Dr. Jekyll, Mr. Hyde, because this is really what these organisms are. And I want to go back a little bit in history. I was lucky to work for a few years as a postdoc with Ignazio Ciappella, who was also a professor at Berkeley, who was one of the key people. He belonged to a key group of people, mostly based in the UK. They actually discovered, but some of them also in the United States and other parts of the world, like Switzerland, they discovered that there was a whole range of organisms that we never knew. And they did that inside trees, because inside the trees, normally we only were able to see the organisms that were actively killing the tree, like the pathogens, or after the tree was dead, the saprobes. But we didn't know that a lot of these organisms, inside the trees, there were a lot of organisms that were totally unknown. And the way they discovered this is by taking branches and identifying fungi, and those bars up on the living branches are the different species of fungi. And then they covered the branch completely, and they girdled it, so nothing could go in the branch from the outside. And all of a sudden, later on, as the branch was being girdled, you can see that those bars in the lower row are completely different from the ones on top, or mostly different. So really, there's a different community that's inside the tree, and this community expresses itself when the tree changes its water content. And this is how this whole research that I'm going to talk about started in the late 70s, 80s, and then early 90s. To be honest, we still don't know how these fungi can be inside plants. I published a paper last year where I was able to find almost 1,000 of these fungi in the wood of poplars, of aspen and poplars in Canada, in a single tree. And that's pretty incredible. And for most of these organisms, again, we don't know what they're doing. But a few years ago, an article came out, which I thought was very inspiring, that shows that these fungi can actually lose their cell wall, and they basically become an organelle inside the cell. And this organelle lies near the membranes or near the vacuoles. And basically, they can utilize the same gene expression mechanism that the plant cell does. So obviously, this cell wall phase, endophytic phase of these fungi is completely different from the phase where the fungus becomes independent. And it seems like that these cell wall mycosomes, which are like protoplasts, they're a little bit like organelles. Think about the chloroplasts, think about the plastid, think about the mitochondria. These are organelles, and basically the endophyte may become a kind of an organelle inside the cell. As I said, we don't know what all of these hundreds of species do, but there is good evidence of many of them have a very beneficial role in plant survival. And in fact, we ourselves in 2019 were able to prove that a certain number of these endophytes were actually preventing infection by a pathogen. Unfortunately, in that case, the pathogen, we wanted the pathogen to infect because it was a biocontrol. And the plant that was resisting the biocontrol because of the endophytes is Myconia caveolens, which is one of the most invasive plants in the world, and is really tragically affecting native ecosystems in Hawaii and in French Polynesia and probably other islands in the Pacific. So one of the things that we know from the very beginning is really the water potential is a huge impact on the growth of these latent pathogens. And what you see in this picture is it's an old paper. This is great data that shows as you go from the left to the right, water is decreasing. So there is less water pressure inside the plant. And those bars that you see are growth rates of this one particular latent pathogen that obviously increase dramatically. So we know that water pressure and pathogenic stage of these latent pathogens are very strongly interconnected. But again, how you change water potential, that can happen in many different ways. And we'll talk briefly again towards the end of the talk. So that's what triggers their lifestyle. But then these are infectious fungi. Okay, so even if they're endophytes, they need to get out of that tree where they're being endophytic, and they need to infect other trees if they want to survive as a species. And they do that on the trees that they kill. Okay, and this is bizarre because normally we have less water pressure when we have drought. So drought really facilitates the growth of these fungi. However, these fungi spread when it rains. And what you see here is different levels of sporulation of one of these fungi. And as you go from the left to the right, it's the exposure to the amount of rainfall increases. So these are actually days of the week. So when it doesn't rain at all, so there's no exposure to rain, there's no sporulation. 2 days a week, there's a little bit of sporulation. Again, 2 days a week, there's sporulation. Then 4 days a week, a lot of sporulation. And the whole week exposed to rainfall, there is a massive amount of sporulation. So again, it's the alternation of dry and wet periods, which really defines California, that is particularly ideal for the completion of the life cycle of these latent pathogens. So infection occurs when it rains. So there are papers out that show that infection of new plants occurs during rainy spells. The source of infection usually is dead plants. So dead plants or dead parts of plants. As long as the plant that's been infected is vigorous, then you have the endophytic life stage. So basically, it seems that these fungi, we're not totally sure that the paper that I presented is one possible interpretation, but it's very convincing. They lose their cell wall and they live inside the cell of these host plants. And then when physiological stress that normally leads to a decrease in water pressure in the tree happens, and also an increase in air and other gases inside a plant because the two go hand in hand. These are aerobic organisms. And so as air inside the plant increases because there's less water and the space is taken up by air, then these latent pathogens start growing. And then again, as they kill the plant where they were living in harmony for many years, they become pathogenic, they kill the plant, then the dead plant becomes a substrate for sporulation. So how did I start getting involved in this? So we have a lot of, there's papers that show the large-scale tree mortality is increasing in North America, and particularly in California. And normally, when we look at these episodes of large-scale or massive mortality, which means the percentage of trees that are killed it's particularly high. Normally, we think of invasive non-native pathogens. So pathogens that come from somewhere else and find the host particularly susceptible because the host has never been exposed to the particular organism, and as a result, it hasn't been selected for resistance. Or we think about complex interactions, so where we can never really point the finger, right? I mentioned insect outbreaks earlier on, but we know that insects are not primary killers. So insects are attracted by trees that are stressed. And this stress can come from wildfires. It can come from high density. That's now come of not logging anymore, as far as they were logged. That's very important. It can come from fire exclusion that it further increases the density. And it comes from root rot. So all of these factors together attract the beetles, and then the beetles are the ones that are, oh, it's a culprit, but really they're rather the secondary. And so really it's a complex interaction that results in these massive outbreaks. Conversely, climate change really is rarely being shown to be directly responsible for large-scale mortality. And the reason for that, I mean, you're CMPS, so you understand this, is that within populations of a tree species, there are so many individuals that are adapted to harsher conditions. And so as a result of that, not every single individual is killed, but you actually have pockets of individuals that survive, besides the fact that you have different microclimates. So there is variable resilience to climate change within native populations up to a certain level. And so how can we, look at these, how can we study these trends in tree mortality? So it's the first, the easiest way in some way is to just do literature searches and see if the reports of mortality are increasing. So these are large metadata analysis. Or if if you're incredibly active, you could have a network of plots throughout California, and you could actually look to see whether disease and mortality are increasing in your transits or in your plots that you've been serving multiple times through a relatively long period. Again, this is, I mean, at the level I'll be talking about, this would be pretty massive, a pretty massive effort. Or there is a third way, which is this is how this research started, which is to look at what owners are reporting. And when you go to diagnose, to see what's going on in different sites, and to try to see what kind of pathogens are coming out of these trees that are being killed. And again, particularly when the reports are novel, as it happened starting in around 2016. And why I was involved, because besides being a professor, I mentioned I am the forest pathology specialist for the entire UC system. And so I actually run a diagnostic lab. And so when people don't want to go to CDFA, usually, or they don't have money to go to private labs, they usually come to me. And so we do the diagnostics on trees that are dying, or even nursery plants sometimes if they're used for forestry or treescaping. But then besides doing that, if something is interesting, we can actually assist with further sampling, and we can actually do pathogenicity tests, which I will talk about. So when you have a novel organism that shows up on a tree, you got to find out whether the organism that you found, is it really a primary killer, or is it just a secondary organism that's coming on the tree that's being weakened by something else? We can do that, which most other labs cannot do, including CDFA. We don't normally do that. And so in 2015, I started receiving reports of relatively large-scale tree dieback coming from different parts of Northern California, and they were from different ecosystems and different plant species. And of course, the owners, often large owners like these big regional parks, or the California Public Utilities Commission, or the state parks, so they were worried because they thought, oh my God, is this sudden of death, or is this something similar to sudden of death that has been introduced, and it's causing now this mortality that we as owners have never seen before. Of course, most of the owners in California don't go back that far. They don't go back that far back, but nonetheless, in the last 60, 70, 80, 50, 40 years, they've never seen that kind of mortality. So that's what kind of piqued my interest. And just to give you an example of what I started dealing with, this was the first case study where I started working. It was pretty massive and pretty impressive. These are blackwood acacias. And this is what one of the sites where we were to look like. So obviously you can see the amount of dieback is spectacular. Not every tree that you see there is dead, and some of these trees actually came back, but I'll show you some metadata in a little bit to tell you what's going on. So this is just to give you an example. This is the worst case scenario, but we're talking about sometimes managers were just telling me about individual trees that were dying, but the numbers of the individuals were high. So you had one tree dead here, one tree dead there, but overall, this was really more frequent than they had ever observed before. And the fact that we were responding to calls is important, because really there was no design in what we did. We weren't looking for anything. We were just responding to owners that were reporting novel mortality in trees. So this is where the power of this study comes, where you can actually get a lot of information without running a massive network of plots across the state. And it's very timely because the reports are coming in, they were coming in, as of 2016. So we started, we realized that it wasn't just the blackwood acacias. And I'll tell you what the trees that were dying. This is very interesting, and it gives you a huge hint of what's going on. And so we wanted to understand how were they dying or dying back, because not necessarily all die, and were there any obvious symptoms? Were there cankers, which are wounds on the tree with the tree responding? Could we see mushrooms or other fungal structures on the trees? And did we find fungi in multiple locations? So was this, if it's just weather related, you expect that trees in different sites are dying because of different reasons. So the biological component of the syndrome comes in afterwards, and whatever is on site that can thrive on an almost dead tree would show up. But if we found the same organism in multiple locations and on multiple trees, that would suggest that these organisms may actually have a primary role, not a sole role, but a primary role in causing the mortality. And then, of course, the big question that everybody wanted me to answer, oh my God, is this a new exotic non-native organism, or are these fungi they are finding native? And so this is a map of the sites that I'm going to talk about. So the trees that, first of all, I can tell you that when we started, the dieback was between in each site was between 24 and 100% of the trees. And we're talking these sites that in the end, when you add up what the sites represent, we're talking about thousands of acres. But if we look at the habitat, these sites represent millions of acres, especially for the native tree species that were being affected. And then in each site, we sampled what may seem a small number of trees, but these trees were dissected from the roots to either the main branches or even the foliage. So each tree was hundreds of hours of work, because we actually went in with chisels, we took all the pieces apart, and if we found any symptoms, so that's the only, we couldn't just sample everything. We were looking for symptoms in these trees. And these symptoms were plated on 6 different media in the lab, which means everything had to be done 6 times. And then we also baited the soil to see if there was a soil-borne organism that was killing the trees using 3 different bait types. So again, we decided to go because we didn't know what was causing this mortality, and it was unreported in the literature. In every single one of these cases, we really wanted to go very deep. So a relatively small number of trees, but with a huge number of samples per tree. And what tree species were we looking at? So blue gums, blackwood acacias, blue oaks, 2 species of manzanitas, California bay laurels, and tan oak. And there is a common thread. You may think it's impossible, but there is a common thread among all of these species. And I will discuss that in a slide later on. So maybe I have to click here. So this is kind of the numbers. And immediately we saw there was a difference between some of these tree species. So in the acacias, which was the first group that we worked on, we could really see symptoms on the trees that were dying back. So we could see cankers, which are this opening on the bark. We could see a lot of fruit bodies on the foliage, but especially the cankers, which we discovered went inside the xylem, inside the wood, were relatively abundant, and almost every tree that had dieback had one of these visible symptoms. And the same thing applies to the native trees that we looked at. So again, different species in completely different ecosystems, but they also had very clear stem and branch cankers that were visible. On the other hand, eucalyptus, which was the one that we sampled the most, we didn't really have any symptoms on the stems and the branches. All of the symptoms were on the foliage, which is, it's kind of what came first, the chicken or the egg, because when the tree goes into incredible physiological stress, the foliage will brown. And of course, we could see the brown foliage and we could see fungi on this foliage, but there was nothing major that would suggest a player that was undermining the health of these eucalyptus trees. So immediately we have two different scenarios. One that's for eucalyptus, and it could apply to many other tree species, and one that applies to acacia, which are blackwood acacias, which are obviously exotic, but also applies to many native plant species. Okay, so and these are the symptoms on, so we worked on 2 Manzanita, 2 Artostaphilus species on Mount Diablo. And the symptoms are here on the right-hand side. You can see the entire plant almost entirely browning. And then you can see this canker. This is what I call a canker. You see it's a lesion on a branch, and everything that's above the lesion is dead or almost dead. And what we found out on the plants that we sampled was we found out three fungi so we found a lot of fungi, but most of the fungi we know after having worked as 35 years as a pathologist, I know that most of those are just pollutants, they're just contaminants. But 3 stood out as being particularly interesting. All 3 of them we know are triggered by physiological stress in general. And the first 2, Neophysicoccus australe and Diaporthe australoficana, they're also known to be latent pathogens in other settings, not in California. And in fact, Neophysicoccus australe and these 2 Artostaphilus species where it was a first report for the species. So this fungus had never been found on this species before. And then we looked at California bay laurels and the trees that were dying. These are the symptoms on the right-hand side. So the entire tree wilts, and a lot of people think it's sudden or death, but it's not. It's actually these latent pathogens, and those are the cankers that can be present both on the stems and on the branches, as in this case. And look at the name. Once again, we found Neophysicoccum, and we found Botrosphaeria, and we found Dothrella. So all of these are very close relatives with a very confusing taxonomy. And in fact, one of the biggest obstacles I'm having in finishing the study is that I have to give a precise name to each one of these players. And the precise name is difficult sometimes to get to because there's maybe one expert in the whole world who knows the difference between one and the other one. And it's becoming, it's really complex. So taxonomically, these organisms are very complex. As you can see, there's multiple of them. One of them, actually 2 of them were major culprits. So these are the ones that we found on multiple sites. And those were Neophysicoccus nuquisitum and Dothrella californica. However, all of them have more or less the same ecology. And this is the key point here. So all of these are those latent pathogens that I introduced at the beginning of the talk. Tinoc dieback in Santa Cruz County, not sudden or death. Again, here we found only one major player. We had 2 players, both diplodias. Again, they're close relatives. We call them bot fungi because the first fungus in this family to be described was called Botrosphaeria. And Diplodia corticula was the one that was, as you can see, that pie slice, the red is Diplodia corticula, which was the most significant. And again, the canopy was drying out. There were other fungi on the foliage, but in each tree that was having the foliage die back, we found these cankers. There were bleeding cankers, similar to what we expect for sudden or death, but it was not sudden or death. And blue oak dieback, this has been reported by a lot of landowners, so many that they told me you gotta come here and see what's going on. And again, the symptoms, so that's a dead blue oak in the front and one that's alive in the back. And that's not a great picture because it's a low dried out, but that's also a lesion on a trunk that we found on these trees that were dead or almost dead. At the time, we were actually almost dead because we don't get anything from dead trees very easily. But these Diplodia corticula, Botrosphaeria, Dothrella were the 2 main players. And again, they're close relatives, and they're very well known to be latent pathogens, certainly facilitated by physiological stress. And then acacia, which was the first one. This was kind of the most complicated one. We found a lot of agents that could be pathogens, but only 2 were widespread, and Diaporthe funiculina and Dothrella viticola are the ones that were really causing a number on acacias. And I like these 2 pictures. The one on the right, you can see how visible these cankers are on the main stem. And the one on the left, this picture, you can see that triangular section. It's actually what the canker does. So it doesn't just kill the cambio, but actually blocks the vessels in the xylem. So eventually, when a significant portion of the stem is being colonized, the entire stem will die. And honestly, if you look at this pie chart without going into all the names, almost every one of these is either drought facilitated latent pathogens, or it's a foliar pathogen, the ones that usually come in at the end when plants are particularly stressed. And here is eucalyptus. And so eucalyptus is interesting because we did find, so when I say other agents here, we did find these organisms that can be latent pathogens, or can, like Neophysicoccum or Cytospora, that are triggered by drought or physiological stress, or Bisconioxia, which is very well known in California because it causes a charcoal canker on oaks. So these are all facilitated by drought and physiological stress. But these were not very abundant. It was one tree that had one of these, another tree had another one. The only thing that we found everywhere was Pseudopseudophila eucalypti, which is probably also an endophyte, but it's not known to be a very aggressive pathogen. So the eucalyptus story is a little bit different, but the origin of the problems that we see, and here you can see the eucalyptus that are basically dead on the right-hand side, and you can see the symptoms on the foliage. This is Pseudopseudophila that's growing on the foliage, and it does grow into the twigs and the petioles. And so we do think it does have kind of a role, but it's definitely a secondary role. So in this case, things were a little bit different, and we think that the primary driver, and we've done a lot more work on eucalyptus, even if I won't talk about it, the primary driver here is the climate change. And in fact, if in San Francisco, I'm working on Mount Sutro, if you just change the aspect of where the tree is growing, you go from trees that are in horrible shape to trees that are doing okay. And that's also why in San Francisco, you have eucalyptus that look much better than in the East Bay, because as we know, San Francisco is cooler, there's a lot more moisture. And as a result, drought stress is only really present on south-facing slopes and on certain type of soils. So again, eucalyptus is a little bit different, but climate plays probably a very important role. And so when I put all the results together, as you can see, the number of colors is very large. There's a lot of organisms that are involved, but the vast majority are either foliar, leaf, secondary pathogens, or they are, in general, there are all of these latent pathogens, and a lot of these are non-latent pathogens, or they are pathogens that we know are triggered by climate. The problem is, well, the interesting thing is also that for each one of these species, we have dominance, so we don't have multiple species that are completely random everywhere, which is what we expect if climate plays a role. But actually, each species has its own major player. And this normally is suggestive of an epidemic stage of a disease. So it means that even if climate means it's triggering, this is really an infectious process. And these organisms are the ones that are being particularly infectious in different type of habitats and or on different species. So the dominant pathogen varies depending on the tree species ecosystem. Even if, for instance, Diplodia curticula, we found it both on oaks and tan oaks, which is interesting. It may be due with the phylogenetic relationship among the hosts. And why do we have dominance? We don't really know. So I don't want to overplay this. So it could simply due to chance. It could be that that pathogen was the first one to get out of another tree, and then it infected one of these trees. And of course, for those cases where we have high density, like those black acacia stands, then contagion would be very easy. One more reason why these super dense stands that we have because of fire suppression and in the Sierra because we stopped logging are particularly dangerous for multiple reasons. Infectious diseases is definitely one of them because of the lack of social distancing. And the thing that was, well, so why is it important to identify each one of these fungi, especially the ones that we know? For us to know that they're major players, we actually need to do all the groundwork. We need to find the same fungus in multiple locations and multiple trees. Well, it's important because there is abundant research that shows that the level of disease, so this picture, this graph on the left, the higher the curve, the more disease there is. So you can see that the curves are very different. And these are caused by the same fungus, but different hosts in general, okay? So different, even if you have the same fungus, the end effect is not the same, but it changes depending on the host. And on the right-hand side, we have the opposite situation where we have the same host, but we have different fungi. And as you can see, there's statistical differences. Again, the higher the bar, the more disease there is. And these differences that may seem small to you in nature, when you have these small differences in the lab, they can amplify, they can make a huge difference in terms of the impact that we see in these different ecosystems. So that's why it's important to know what's there and to understand the different combinations. Any of the things that we found alien, so non-native, yes, but the reality is that these are organisms that come with the plant. And these were unreported. These particular ones we know are not, they're not native to California, but they've been reported in other places where the same plant is being planted. So it's kind of like your microbiome. People go everywhere, they bring what's inside of them. So yes, they are non-native, but these are not, necessarily highly infectious organisms that have been introduced. They are organisms that are introduced with the plant, and they're now expressing themselves because of physiological stress that's affecting these plants. That said, when I piled up the results of the first 3 or 4 years of work, I found 12 first reports. So these are 12 first reports at the worldwide level, or for California, or for the host. And I don't want to go into the details. And this is super exciting when you're a scientist. You find one single study, you find 12 things that nobody has ever reported before. But it's a huge mess, because now I have to prove that these really are aggressive pathogens, and they're not just secondary organisms. And you know how difficult that is. I mean, if you remember what happened with HIV and AIDS, how difficult it was, the same applies to these trees even more. The trees don't go to see the doctor. And so there is a lot of work to do. So I was excited, and then I said, oh no, now I got to do the work. I actually got to prove that these are real pathogens. And so how do we do that? We do that by running pathogenicity tests. This can be done on trees that are in the field, or more commonly, they're done in potted plants, sometimes pretty large in the greenhouse. And what you want to do is we put the pathogen in these. For instance, this is the acacia study, and then you compare what happens to the plant that are infected by different of these putative pathogens. And you compare that disease level with the controls, which are not they're uninoculated, but they're wounded in this case. This is important. So this tree is wounded, and this is the dam on the left-hand side here. Again, the higher the bar, the larger the lesion, the canker. And the lesion that you see in the controls is simply caused by my wounding the tree. And so as you can see, without going into much detail, I was able to prove that Diaporthe, in this case, Diaporthe funiculina, is a very aggressive pathogen of acacias. And so now I had to repeat it not for every single one of those 12 first reports, but for the major ones, I had to repeat the work. But I was more, that's kind of a repetitive work. So I was actually more interested in, now that I know that it's a Diaporthe, and now that I know that, let's just focus on Diaporthe. Now that I know that Diaporthe is a major player, and because I know that Diaporthe is a latent pathogen based on the literature, can I show that drought is actually exacerbating disease? And this is where the line of work that I've been doing for the last 3 years, including all your sabbatical, started. The results were very encouraging. So here on the right-hand side, you see 2 boxes. Again, the higher the box, the more disease. Letter C is, they're statistically different. This is the lesion caused by Diaporthe funiculina, multiple plants, in the absence of water stress. And D is the amount of lesion that's caused in the presence of water stress. So we basically drought stressed these plants. So this was done obviously in the greenhouse. And as you can see, you get much bigger lesions, but also you get less variance. So you really are causing every single individual suffers severely because of the presence of this particular pathogen. So this is consistent with this, issue of the mega drought going on in California. And even if it rains, once you have multiple years stacked up of drought, 1 year of rain, 2 years of rain are not resolving the problem. So the mega drought starts, it's a problem that it's much more complex than just having 1 or 2 rainy years in between. And drought in this case really exacerbates the disease, or at least the pathogen. So we started to understand that climate had something to do with this novel mortality that was being reported across California. And then we repeated this for the other 3 species. And what I wanted to show you here is the blue oak situation. And again, in this case, we used Botrosphaera dothidia, which was one of the 2 major pathogens we found. And again, you can see the difference between drought and no drought, okay? So again, and then you can also see what happens when you don't really have a pathogen. In the middle there, those 2 bars, independently of water stress or no water stress, it doesn't really matter. The lesion is fairly small, although it's not nothing. And in the case of the controls, statistically, the two lesions are basically the same. So we were able to prove again, and here on the right-hand side, you see a small lesion on a tree. That's how we do it. So we inoculate, and a few months later with the bark, and we measure. You can see the dark area in the middle. We may sometimes it can be very large. So again, we were able to show that drought was really playing a role in exacerbating these kinds of diseases. And even if we had different species, all of these species are phylogenetically related and ecologically related because they all have the same ecology. And then again, this is Coast Live Oak, another example. This is an example that's striking, even if we didn't really work on Coast Live Oak. I just added this for fun. And I found out that the same pathogen we tested on blue oak is really aggressive on Coast Live Oak, but only when there is water stress. So again, the first and the second column are lesions on Coast Live Oaks caused by Botrosphaera dothidia in the absence of drought and in the presence of drought. So I found this very interesting. And so based on the results that I obtained on Coast Live Oak, and then based on the results that I obtained from maples that were dying, I developed this concept of the environmental disease component index, which determines whether the disease environmental stress can actually increase pathogenicity. And so I'm not going to tell you how this is calculated, but if it's over 1, it means that the environment really affects the disease. And if it's over, if it's positive, and if it's over 0.6 or 0.8, it's really significant. When it gets to 0.9, really the environment drives disease expression. In this case, it was 0.91 for the fungus Botrosphaera dothidia on Coast Live Oak. And so once you see the arrow there, that's the beginning of really pretty huge impact that the environment can have on the disease. And I repeated this for the other ones. And what happens is that some of these fungi have a big impact. And so basically, this is kind of a schematic representation of the effect of the physiological stress and the effect of the pathogen. And it's not based on real data. I haven't finished this yet. But as you can see, these different combinations populate different parts of this graph. And the more you are high up, it means that you're more aggressive as a pathogen anyway. And the more you're on the right, it means that the environment affects you significantly. What is correct here is the position of the blue gum pathogen, which you can see is really low in the corner. So it means that it's not an aggressive pathogen anyway. And this is based on experiments that we did, like the ones that I've just shown you. And even when we subjected the eucalyptus to stress, this particular pathogen didn't become more aggressive. So this is why I have data to show to prove that what's happening to eucalyptus, it's really mostly due to the fact that eucalyptus doesn't belong to the sites where it was planted. And it was okay 50, 60 years ago, but unfortunately, because of climate change, that's not okay anymore. So basically, every single owner that came to me since 2016 that I decided to pick up the research, all of the dieback, not necessarily mortality, was caused by latent pathogens. And again, most of them, most of them in these 2 families. There's a 3rd family that's Xylariales that's also important. So this was surprising to me. And I'll tell you why, because normally these latent pathogens, until a few years ago, they're kind of exceptions to the rule. And I'll show you one of these exceptions. Like you find them on like redwoods that are planted in the Central Valley, right? So things like that. So something that where you don't really know, is it really is the pathogen, latent pathogen, or maybe it's the tree so offsite. But in this case, the abundance of these latent pathogens all of a sudden suggests that the driver is really affecting the ecosystem at a scale that was unprecedented before. And we have multiple of these. So not just one gets favored by stress. So you have multiple things growing even on a single tree, but we do find some that are dominant on the tree species and geographically. They're not the only thing by any means, but so that means that something is going on in terms of the infectious process. And we were able to show experimentally that water stress is increasing disease. And we also know that for some trees, for instance, for eucalyptus, climate may have a huge role. And for the other tree species, the pathogen really has a bigger role, even if it's triggered by climate. So that means that we were able to prove it with the experiment I'm going to show next, that harsh climate doesn't kill the trees at this level, not yet, at least. But when you have the pathogen does kill the tree. So it actually is the pathogen that's killing the tree. It's not the climate. So I was able to show the drought affects the health of these trees, but especially increases the severity of disease. And then I moved on. I went on sabbatical and I met Vladimiro Warnacha, a professor in Torino, and he was working on these latent pathogens in agricultural settings. And so I was interested in working with him on just looking at the effect of temperature. And this is how the experiment was designed. So these are different, either drought-driven or climate-driven, physiological stress-driven fungi, almost all of them. And these are their optimal temperatures in vitro. So how well do they grow? And I kind of highlighted two of them to show you that this optimum temperature can be quite different. So, so what happens if, let's say, the temperature goes up, from 25 to 29 degrees? Which one of these two fungi, Bochosphere dothidea or Diaperthe rudis, do you think it's going to be more affected by disease? Definitely Bochosphere dothidea, because if you go up from 25 degrees, you actually get to the optimum growth. In the case of Diaperthe rudis, that wouldn't work, because if you're over 25, you actually are where the fungus cannot grow anymore. And each species has a different optimum. And so we started thinking that, oh my God, Bochosphere dothidea likes warmer temperatures. So if I have more days that approach 27 degrees, I may have more disease. So climate, just warming can really affect disease. Yeah, it's a nice idea, but you got to prove it. And not many people have done it. People have done the drought, but not the temperature. And so we picked a system that he had worked on, which is blueberries. And I like blueberries because it's in the same family as manzanitas. And the same genus that we had on manzanita, Neophysiococum parvum. And here on the left-hand side, you can see the growth rates with temperature of this fungus. These are in vitro. And as you can see, the fungus really thrives at a relatively high temperature, 30 degrees. And so the more days you have that approach 30 degrees, the faster the fungus grows. Well, does that affect disease? And so these are disease levels. So this is a little different from the way you've seen this before. It was on the y-axis disease. In this case, it's on the x-axis. So the more you are on the right, the more disease you have, okay? And the higher up you are, the higher the temperature. So you can see the Neophysiococum parvum that does its best in vitro at 30 degrees causes significantly more disease. As you can see, the letters indicate differences, statistical difference when the temperature approaches 30 degrees. So we're actually able to prove that because the temperature is more favorable to this particular fungus, this fungus causes more disease as the temperature warms. This is actually lesion sizes in plants that were inoculated by Neophysiococum parvum and kept at different temperatures. So then we modeled mortality, not just disease. Here we did a full experiment and we modeled mortality. And this line here indicates the probability of death of a plant. So as you can see, when you stay at lower temperatures, 20 Celsius, as you start 25, as you arrive at 30 or 35, the probability of mortality goes up almost to 100%. And this model was based on actual mortality rates that we obtained. So these are mortality rates on the right-hand side. As you see, we're back to the normal way of showing things. The higher the bar, the more mortality. So you can see that as we reach 30%, you really have a very high level of mortality compared to other temperatures, even not that particularly different from 30 degrees Celsius. So we were able to prove that mortality really increases and symptoms increase with temperature because the organism likes warmer temperature. However, we were also surprised because basically what we proved is that if the pathogen is thermophilic, or even if it likes warmer temperatures, if the temperatures warm up, that warmer temperature would increase the growth rate of the pathogen and will also increase disease and mortality in plants. However, if we look at how much more mortality did we get compared to how much more growth we get, we actually get much more mortality than, and much more disease than the growth rate itself. So what it means is that what we see is the combined effect of the increased growth of the pathogen. So the pathogen is growing more, it's like cancer that's growing faster. But also there's something else that's not explained by that. And we think that that's explained by the physiological stress that's imposed on the plant by a higher temperature. So actually the outcome of the temperature study was pretty definitive and was very interesting because as temperature increases, the increase in disease is not linear as we expect, but it's kind of exponential because there are two different things. One is the fungus is happier, and the other one, the plant is less happy. And then I should have finished here because beautiful studies, drought works, temperature works. And I said, no. Now I want to do, again with Vladimiro, we're kind of done with that study. And I'm like, yeah, great. We were able to prove it. I'm one of the first people to do so. But I want to put together temperature and water stress in the same experiment. And this is where things get really complicated. So we're able to get clones, so plant that's clonal. So we actually had no variation. And we used Mimosa Acacia dealbata. We used the Aporta funiculina, which we know is important in California. And we do have some Acacia dealbata with the Aporta in California. We had different treatments. So one was wounding, because remember, wounding is necessary for me to place the pathogen on the tree. Then we had plants where we didn't do anything. And then we had pathogens where we wounded the pathogen and we also put the pathogen. So we had 3 different treatments, 2 different temperature, relatively cool, 22 Celsius, and pretty warm, 27 Celsius, water stressed and not water stressed. And we ran this experiment between the end of November, mid-February. And we looked at symptoms and mortality. And here is these are these plants, the size of the plants. And as you can see, they're fairly small. And this is on the right-hand side, it's a plant that was inoculated. So we cut it with a scalpel a little bit, and then we put the pathogen. So obviously, on a small plant like this, we expected wounds to be relatively significant. And they were. And also the way this experiment works is different. We don't look at lesion size. We simply look because the plant, this is what we picked it. The plant is all or nothing. So the entire plant becomes symptomatic, and then the plant dies. So it's a 0-1, which makes, when you have so many plants, it makes the measurement a little bit easier. So the experiment went on for several months, and the plants were analyzed every 2 days. And they were watered every 2 days, or water less or more. And you can ask me how we did that. But basically, we wanted to make sure that we were above wilting point for the plants, and that the plants also were a field capacity for the soil, which means very well watered. So when we analyzed the entire data, we were very happy because we found out that our expectations were met. So this is the entire data. So it includes plants that were kept at the different temperatures and different water regimes, but overall, the data has a meaning. So when you have the fungus, you get the highest mortality. When you just have the wound, you get intermediate mortality. And when you don't do anything to the plants, you have a little bit of mortality. And you know what? The conditions were pretty harsh, and we went on for a long time. So when you keep plants in drought conditions, just above wilting point with a high temperature, you would expect eventually that after a couple of months, plants start to die. That's what happened. So this was very consistent with what we expected and was a good sign. And then, of course, this is also to be expected, but I have to show it to you what happens through time, because this is the problem of statistical analysis of experiments that are where the measurements are repeated through time, but you're looking at the same plants. So every time you're looking at the same, and here are different times that are actually statistically different from the beginning to the end of the experiment. And the black bar is the number of plants that were symptomatic. So as you can see, as time progresses, we had 3, 4, 5, 6, 8 time periods from the beginning to the end that were statistically different. And every time period, the amount of symptoms increased and also the amount of mortality increased. But I'm just showing you the symptoms. And this is the data set that we have to analyze. As you can see, very easy for me to talk about this data set. So this takes into account everything. Time, so when you go from left to right is time goes by. And then you have drought and you have high temperature, you have wounded plants, you have plants that were just left alone, and you have plants that were wounded and inoculated. And of course, I can't talk about this data like this. So what I'm doing, I divided this in 5 different periods where the interactions were significant. And I will give you a little snippet for each. So remember, 1 is the beginning and 5 is the end of the experiment. So at the beginning of the experiment, we actually had a few plants that were wounded that died, whether they had the pathogen or not. And that makes perfect sense. And we always get that. So when you have small plants like that, it's a reaction to the wounding. And so you have, so the only significant variable statistically was wounding, not having the pathogen or not having the pathogen. And a very small number, so it didn't really fit the experiment. But that's the first thing that you see, a response to the wound, which should make us think about what happens when there is mechanical damage on plants. In the second phase, having the pathogen really causes more mortality, and more so at the warmer temperature. So basically, of all the different treatments at the different temperatures, it was the pathogen inoculated plants at 27 degrees that had the highest mortality. So if you have the pathogen, water stress at the beginning doesn't really make that big of a difference. It's really temperature that's driving mortality. And this was very interesting because I just published a paper about flooding and pathogens, where we actually were able to show that plants that were infected by a pathogen resisted flooding better because the plant that's infected responds to infection by closing the stomata. And so there are some things that the plant does that make it actually more resistant to physiological stress, serendipitously, because it's responding to the pathogen. So, and we were able to show that because the plants that were mock inoculated at warmer temperatures, but with the water stress, had also a very high level of mortality. So again, pathogen and warm temperatures and warm temperatures and no water cause the highest level of mortality. As we move on in time, number 3, that's the third phase, the presence of pathogens is the most important factor. Then temperature, and we can actually rank these statistically, and then water stress. So later on, water stress becomes important, but is actually less important than temperature, and it's less important than having the pathogen. So, but of course, when you have the 3 together, then in the middle phase of the experiment, the highest mortality was in pathogen-inoculated plants at 27 degrees and with water stress. But again, there is a rank in the importance, and water stress comes after the temperature, which was a surprise for me. And then towards the end of the experiment, then water stress becomes really important. So water stress becomes a driver. So in prolonged periods of time, it's drought, and that's why the mega drought really has this huge impact. And then temperature becomes insignificant at that point. But remember that the survivors are very few at that point. And then at the end, even the plants that are untouched, so only at the end, the plants that were left alone, they started dying if they were water stress and if they were at warm temperature. And in this case, water stress was actually more important. So we were able to show that we were using very harsh conditions, and at the end, even those conditions were starting to kill plants. But again, it was at the end of the experiment, and overall the numbers were pretty low. So what does this mean for us? So really, what are we going to, what do we expect when we have these latent pathogens? We expect that as the temperatures warm because of global change, we're going to certainly see more disease. And then, of course, that disease is going to be compounded also by water stress. But water stress by itself, without the warming, is not as powerful. That's kind of the take-home message. And it's kind of interesting that actually the pathogen may help with water stress at the beginning, but that doesn't last, as we've seen in the previous graph. I mean, on the previous slide. As time goes by, then water stress becomes also a significant factor, even if it's less important than the other 2. So in this case, I was thinking, oh my God, maybe it's the same, we're looking at the same thing that we looked at in the previous paper. Temperature really is important because the pathogen likes warmer temperature. And so we did a growth rate study, and these are growth rates in vitro. And I can tell you that the only strains that killed more trees at warmer temperatures, we had 4 strains of Diaporthy. And this is why the study is so complicated. But the top one that you see here was aggressive no matter what. So it was really killing a lot of plants even at 22 degrees. And statistically, the number of plants killed at 22 and 27, this is growth rate. But when we looked at the number of plants killed at the 2 temperatures, but this strain only, it was statistically identical. However, when we looked at the other strains, the ones that are below here, those were the ones that caused different levels of mortality statistically at 27 degrees. But look at this. Temperature is not any affecting the growth rate of the pathogen. So this was important because this is a different case than the other one that we had studied with blueberries. So in this case, the temperature is not directly affecting the, beneficially affecting the pathogen. And so basically, we were able to show that the temperature is mostly affecting the physiology of the plant, which is very important for us to understand. So it's the temperature affects the physiology of the plant, and the change physiology triggers higher pathogenicity of latent pathogens. The pathogens actually can help at the beginning with water stress, but that doesn't last particularly long. And we do reach, in the end, we do reach maximum symptoms in plants infected at a higher temperature and at a water stress. But again, it really is a physiological effect of the plant that triggers the pathogenicity of the fungus. And also very important, the drought by itself it's a secondary driver of disease, but it becomes more relevant at higher temperature. So having a drought, and it makes kind of sense, obviously. If you have less water and it's warmer, then you suffer more. And again, it points to physiological stress being really relevant. So again, I hope I made myself clear. It's very complicated, but so I think I made the case that drought is really affecting our California trees and making these pathogens more aggressive. Temperature is making some of these pathogens more aggressive, the ones that like warmer temperature. But overall, temperature and water stress, increasing temperature and water stress are bound to really trigger a huge outbreak of these latent pathogens. So Dr. Jekyll, Mr. Hyde, can we go back to, can we reverse to being benign again? Yes, but there is a threshold. And is the disease, can we really deal with this disease? No, we can't. And the reason for that, not very easily, we can only deal with the disease by making sure that plants are happy. So every time we plant a plant in the wrong place, the plant will be unhappy. And these diseases now are becoming prevalent because of the stress that's caused by the plant by climate change. And we've been able to show with the experiments that climate change by itself at this point is not enough to kill the hosts. So the latent pathogens really are players, and they've been underestimated. Why? Because as I was telling you earlier on, these pathogens originally were considered an exception. So is this really what's happening? So this is an example of the threshold. So this was an experiment done in the 80s. And on the left-hand side, these are weeks. And if the stress goes on past the orange line, there's no reversal. But if the stress ends before the orange line, then there is reversal. The problem is that line will be different for each one combination of latent pathogen and host. And as you've seen from my first results, there's so many combinations. So this is very difficult to predict. But we do know there's a threshold after which there is no return. And I am afraid that we may be close for some plants to that. The ones that I studied were very important model plants: blue gums, acacias, bay laurels, blue oaks, and manzanitas, and tan oaks. And I'll tell you why in a couple of minutes. This actually is the result that I color-coded for you of what we observed 4 years later, or 3 years later, when we went back to the same sites. And if the line, if the row is orange, or red, things were worse. If the line is yellow, things were better, but only because the trees that were dying were taken out. So we don't really know. We can, and if it's white, we couldn't say anything because all of the trees had been taken out. So the white rows, the trees were taken out. And the reason why they were taken out is because they were very sick. So that basically means things got really bad. So really, are these trees going back? Remember that we had the second sampling happened in the fall of 23 and in the spring of 24. So we were after the rains of 23 and after the rains of 24, or during the rains of 24, and things didn't change. So actually things in general got worse. So again, this is, for me, now that I've looked at these trees for multiple years, I am a little worried because I thought things would go back, but obviously these are the sites that we were studying, which may not be representative because these were chosen based on what the landowners were telling us. And so this may be the worst case scenario, but the worst case scenario is already happening. And the average amount of dieback across all of California, the studies that we studied, the sites that we studied, was almost 70%, so 68.4%. So that's the average value. It's a really high value. So this is really affecting them. If it's not killing them, it's really making them miserable. And I will skip this one. These are some pictures of symptoms again, and that's a schematic representation on the right. The slide before was to tell you that these latent pathogens can be very fast. And this is just data that proves it. But why it can be very fast? Because they already are in the tree. When you contract a disease, you actually have to contract the disease, and then the disease has to become established in the host. In this case, the pathogen already is in the host. And what happens, the blue area here is a canker, and those are the starting points. So when you have endophytes, you could have multiple starting points of a canker. That's on top, it's the normal canker, the garden variety. But with the latent pathogens, you could have multiple starting points because you have multiple cells that are being colonized by the endophytes. And then together they coalesce, so they can cause much greater symptoms much faster because of that. And on top of that, they're already inside the plant. So this is the kind of the epidemiological life cycle. And I would like to stop on the first slide, which is a drawing that really, where do we see these latent pathogens? And I'll show a slide that really points out the point. We see them on exotic plants that obviously are planted off-site. And we see them on native plants that have become invasive, mostly because of fire exclusion or because of grazing practices, California bay laurel. So these were usually California bay laurels that are naturally growing, but they're growing not in the riparian areas where they should be growing. They're growing everywhere because of fire exclusion and grazing. And as a result, these bay laurels in these areas that are far away from water sources, in less mesic areas, they're becoming very susceptible to subtle or climate change. Or we're finding the disease in areas where trees are already living at the limit. Manzanitas in particular soils of Mount Diablo where it gets super hot, and blue oaks, which we know that area of the hills is particularly difficult. And we have oaks and we have grapevines that really are the only ones that can become adult trees because it's a stressful environment. So really there is a common thread and it affects all these trees. These trees are either off-site because they were planted there, they're off-site because they become invasive, or they are very susceptible like tan oaks, or they're living at the edge already, like blue oaks and manzanitas. And this is kind of the drought causes these lesions to develop, mortality, and then you have sporulation on the dead trees, and then you can actually get large-scale mortality because the trees are subjected to the same stress at the same time. Sorry. And originally, I got involved with this type of fungi a few years ago, and this is what was the norm. So we reported a canker of bristlecone fir. This was just a massive canker, several trees that these massive cankers. Where? In the East Bay Botanic Garden. That's where the arrow points. And look down, that's the native range of Bristle fir. So obviously the hills of Berkeley are absolutely off-site for this tree. And that's, oh yeah, we have these latent pathogens because the tree is off-site. But now even native trees in their native range are becoming off-site simply because they can't move that fast. And so these latent pathogens, as I've shown you with my data, are popping up everywhere. And they can be very fast because they're already inside the host, maybe doing something good. But all of a sudden, from Dr. Jekyll, we become Mr. Hyde. And because of climate change, these are going to become major players because eventually climate is going to make plants very unhappy. And that's exactly what these latent pathogens need. And of course, if you are at the edge of the range, or if you are in a very harsh habitat already, you're going to suffer first. So that's what we're going to expect. So basically, these are the pathogens. It's not going to be sudden or death. These are going to be the pathogens that are going to reduce the range of our native plants by starting at the edges where the plants are already a little bit borderline. And now with climate change, things have changed dramatically. And so this is the common thread. Exotic trees that are, and I could talk about acacias and eucalyptus, but I can tell you that both of these species, I went to Australia and Tasmania a few years ago, and both of these species come from southern Australia or Tasmania. Originally, they are not drought-loving species at all. They just survived in Northern California because of a complexity of situation, but they're definitely off-site. And native mesic trees that are becoming invasive like bay laurels or even tanox, because tanox in the absence of sun or death can be kind of invasive. They're too dense, too many trees, and they're growing in areas where originally they were not growing, and they're becoming a lot more susceptible. And again, we do have species that already are at the edge. They're at the edge, and they're at the edge. And this change is causing these latent pathogens to take them out. Again, it's not a climate. It's the latent pathogens just taking them out. And this is my last slide. Actually, 2 slides. I also wanted to point out that water shortage, which we know it's important, it's caused by a variety of factors. One of them is increasing temperatures, which you see on the left. On the right is increased variation in rainfall. It's actually exactly what these latent pathogens love, because they love alternation of very dry and very wet periods. So when you have this alternation of very dry, very wet periods, it's absolutely ideal for these latent pathogens. And on the right-hand side is the number of fog days, which is actually decreasing. And if the models are predicted that they would increase, they are decreasing. And obviously, as we have less fog, there is more stress. Everything equal with less fog, trees will require more water. Fire exclusion is having a huge impact because of density. So high density of trees already, besides all the other things that can happen after a fire. But even in the absence of fire, fire exclusion puts a huge load on the ecosystem because the densities are too high. And the same thing for logging. And I work on root diseases in the Sierra Nevada, and I can tell you that in the absence of logging in forests that were logged significantly before, we have a dramatic situation of super high densities that makes these pathogens particularly abundant, even if I haven't studied it yet. I studied root diseases, and root diseases are really more abundant in these unlogged dense stands. And then, of course, planting these plants where they don't belong, which is what we have done in the past. And so really, the only way to get to how do we deal with these pathogens? So there are 3 possible scenarios of what's going to happen. One is, number one, which is the worst possible. It may happen for some of these tree species, that we go from having a lot of species to having none. So mortality, this could be mortality. So we go from 0 mortality to have, really high levels of mortality. We have number 2, which is a scenario where we have mortality, but then things get better again, and we go back to the baseline. So no mortality. What I think is most likely is actually number 3. So mortality goes up, and then we may go down, but as you notice, it doesn't go back to the baseline. It stays up high. So there is a trend that eventually is going to be similar to the scenario number 1, but it's going to happen cyclical. And I'm pretty sure that this is probably what's going to happen, but we still don't know. We don't have enough data to predict. And again, what can we do? It's very difficult to deal with these diseases because these latent pathogens are part of the microbiome, and in fact, they may be beneficial. So they're already inside the tree, and they are infectious. So as the trees die, those dead trees are the substrate for the next generation of infections that's going to affect more trees. So really, the best thing we can do is to make trees happy. So we made this, so either we plant trees of a species that come from southern locations in the north, or especially in the case of exotics, we should get rid of exotics and plant trees that are ecologically not adapted to Northern California, but probably to Southern California. So we gotta start thinking about that we may benefit from stopping these outbreaks of mortality, because these outbreaks are really responsible for potentially massive infection. And we think that's what happened with the acacias, because the acacias are very dense, and the outbreak was really incredibly, as I've shown you that picture at the beginning of the talk, incredibly significant. So making trees happy could also mean thinning our woodlands, putting trees back where they belong. So let's avoid bay laurels from proliferating where they don't belong, as an example. It becomes more difficult when we have trees at the edges of what they can do, like potentially the blue oaks and some of these manzanita species. I don't know that there is an easy solution in that case. But in agriculture, they do remove the dead plants to prevent infection. So in case of really high value situations, or where some species may be at risk, some populations may be at risk, and if it's feasible economically, maybe removing the trees that are killed by these latent pathogens, or putting the dead branches, the dead stems under in the soil. They're not very good competitors in the soil. It could be a partial solution, or could slow down the process. So unfortunately, this is all that we have. We do know that the genotype of plants affects the endophyte, not just the species, but even the genotype. So we need probably long-term research to see if there are some species that are more, once we know the major player in terms of pathogen, we can find plants that are maybe more resistant to infection by the major player. But eventually, it's these latent pathogens are generalists, and so it's possible that there's going to be another one that pops up on a species. But at least knowledge of what are the major players may help us in that research. But again, we should be careful, because we may have oaks that are resistant to Diplodia corticula, but they become susceptible to Butyrosphaeria. And so I'm not sure that that's a really solid solution for the long term. It may be a solution for the short and the midterm if we can find those trees. And with this, I conclude. I wanted to let you know that we do a lot of public education. I run a large citizen science program that's called SODBlitz, because sudden or death is still very important. Even if the drought is slowing it down significantly, it's still a major player, and it can come back really fast when we have rain. If you feel like you want to help, I train more undergraduates in my lab than any other lab in my college probably. So if you want to help the undergraduates, you can make a donation and write undergraduate. It's tax deductible, and you can find the link on my site, just in case you feel generous. And I will take any questions.
Wow, that was incredible. Thank you so much. Terrifying and incredible. We do have a question about which park you were talking about in the East Bay from Robin Mitchell.
Okay, well, we have multiple locations. If you look at the map, you can see them. Chabot is one. Leona Heights is, the picture was taken at Leona Heights. So it's not just one, it's multiple. Carquinez Strait, and we give different names to the parks. Well, if I go to the beginning, maybe you can see where the dots are, but it's hard because they're the scale. And we worked in the SFPUC area there, so you can see on the East Bay all those little dots. Sometimes they're overlapping.
Which PUC sites do you know?
Yeah. Crystal Springs, both the higher and the lower reservoir.
Wow.
You can see the dots there. Those are actually multiple dots, but.
And then we've got some really good questions from, and I'm going to mispronounce this name, Ore Karmi. Is it all right if I permit this person to talk rather than?
Absolutely, yes.
Okay, great. You are.
Hi, can you hear me?
Yes, I can.
Hi, thank you for this talk. I've heard you multiple times and I absolutely love all your talks. But I had three questions that I wrote. One was about the pathogens. Are they naturally intracellular, or is it just in the pathogenic stage that they become intracellular?
No, so they would actually be intracellular in the endophytic stage. So they actually need, and remember, this is one hypothesis. So they need to be intracellular. It's based on this hypothesis, which I really like because it explains so many things, but not everything. They actually, in order to be endophytic and maybe don't do very much, they need somebody else to do the transcription for them, right? So it seems that they can have the plant code for some of the genes they actually need. By they become part of the cell. And this paper that I cited was fantastic because they were able to do that. So in the lab. So it's not just an idea. They actually took a fungus, we took the cell wall off, we put it inside the plant, and the plant started coding the genes from the fungus.
I see.
And then when they become pathogens, they maybe the rupture of the cell and the presence of air, because remember, they're aerobic. This is key. They're aerobic. So they need a certain level of oxygen. And so it may not be necessarily be the water stress per se, but it may be the increase in oxygen and CO2 that happens when you have less water in the plant. And so then they start growing and they grow intercellularly, but then they will kill the cell and eat the nutrients from the cell. But most of the growth would be intercellular.
I see. Thank you. The other thing I might have missed this, but did you take measurements of the temperature and water stress in the field just to correlate that with what you were finding in the lab?
No, we didn't, because these are regional trends. I mean,
Okay.
we could just look at the regional trends. I mean, there is data available, and I've shown you a picture with increasing temperature. That was for Northern California. So the data that I've shown you in the final slide was, so we know it's happening. We know it's happening. What I did notice was in San Francisco, we work in Mount Sutra on eucalyptus. And I noticed, it's so clear that it's the south-facing, the trees that are in the south that are really suffering. The ones that are west-facing and northwest, they're still doing okay. They're not perfect, but they're doing really quite well. And but in the East Bay, it's different because obviously the difference between San Francisco and the East Bay is enough to make the symptoms much more widespread in the East Bay, independent of the aspect.
And my last question is kind of similar, and maybe it's me just denying how, and I'm refusing to accept like how significant the climate change has been recently. But it just seems it's hard to accept that all of a sudden, like all these different organisms of different organisms taking over a different tree and cause problems all at the same time, and so I'm just kind of wondering if there's more that you can say about that.
Well, I think it's a perfect storm because, okay, so, when people from Australia and Tasmania come, they will say blue gums here, they already used to say that 20 years ago, okay? But they didn't think that the Bay Area was good for blackwood acacia, and that was a long time ago. And in the change that we've had, and then I'll make another example. The change that we've had, so you have to understand that it's all the things that I talk about at the end of the talk. So increasing temperatures, less fog, which remember, it's really important for the Bay Area. Yes. And then a lot of more variability in rainfall, right? So we get a lot less rain and then a lot more rain just 2 years apart or a year apart. This is kind of, and high density. I mean, look, that blackwood acacia stand. So remember, the density increases with time, right? In an unmanaged stand. And so the densities that we have now are much, much greater than the densities that we had 40 years ago. And if you look at the hills that are covered by woodlands now, even 40 years ago, they were less covered by woodlands. So there are changes that we can visually see if we think about the last few decades. So I think that each change per se would not have been enough, but when you put all of them together, it's definitely enough. And the bay laurel story is very interesting because, again, how long does it take for a bay laurel to proliferate and go from its real habitat to a non-habitat because of fire exclusion? Not that long, 20 years. And that has really happened now. And, Joe McBride at Berkeley was the first one to write a paper about it, about all these bays coming in because the cows were grazing the oaks, not the bays, and because of fire exclusion. So in the last 20, 40 years, I think we have started to see a peak of these ecosystem level changes. So it started 60, 70, 80 years ago, but there's resilience in the system. But now we are at a point and where things have changed, and the densities is particularly where we have these densities. I think it really worsens things because of the toll on the water resources, and also because of the infectious stage of the fungus. Because when you have trees that are next to each other, they can infect each other very easily. So it's kind of a double whammy. So again, I think this is a perfect storm. While for blue oaks and manzanitas, I think these are plants that really live in harsh environments. Hey, you increase the temperature by 3/4 of a degree, and you take away maybe 30 days of fog for Mount Diablo or 10 days of fog. That could have really major impacts on the ecosystem. So I agree with you that, and maybe we don't perceive that because we live day by day. We don't perceive that it's the synergy of all of these things together and the bad management, because planting these trees, of course, you're CMPS, so, planting these trees doesn't, and people say, oh, but we love these trees. But I tell them, yeah, I know, but we gotta be strong. They don't belong here, and we better take them out ourselves rather than waiting for them to die en masse, create a fire hazard, and infect other trees, including native ones. Because remember, again, and this mortality that I'm talking about happens in pockets. So it's not like the entire, it's not taking out the entire woodlands in the Bay Area in Northern California. It happens in pockets, but it's becoming more significant. And the problem with these endophytes that turn into pathogens is that all of them are generalists, okay, with preferences maybe, but I said the preference may be historical, right, just because of opportunity. And so the problem is, if we create a huge amount of spores, of inoculum on these eucalyptus, the spores may end up on coast live oak, and all of a sudden we'll have a new of these latent pathogens that it's maybe more aggressive than Diplodia and Botrosphaeria. Right. So I think that's kind of, I don't want to be, again, this is not doom and gloom. I'm trying, and I really want my data to help us make decisions for the future. I'm not saying these are going to take out all of the trees, in the next 10 years, but they're starting to erode the integrity of the system, and they're starting to erode the diversity of populations, right? So they're starting to do, and it's a good thing that it's really major on exotic plants or on plants that are off-site, but I think it's kind of telling us in advance what's going to happen, right? Because but we don't know how fast it's going to go, and we don't know the resilience of different tree species and different systems. So again, I don't know. It could be faster than I think, or it could be slower, and it could be that every tree species is going to have a different rate. So it's insidious because it's happening, but it's not like bark beetle, outbreaks, which you see them all at once, and then you think, oh my God, but then those trees come back, no problem. So it's in this case, these trees may not be able to come back because you're going to have a resident population of the pathogen ready to infect them. And if they're unhappy, the pathogen will thrive. So again, making trees happy, silviculturally or by planting the right tree in the right place, even the right, genotype or the right provenance. Again, I really think that taking trees from the south and planting them in the north in areas that are suffering already may be a good idea.
I see.
Maybe that would be something that I would encourage, and the problem is if we wait, oh, I'm not going to do it until I see the trees are dying, it may be too late.
Right. I guess the one part, now that I hear you talking about, the one part that I have still difficulty with it's happening to multiple tree species all at the same time. And I guess in my mind, it feels like different trees are going to have different um, thresholds, so why
But
is it happening to
yeah, but
multiple?
besides the perfect storm, what you have to think is that we don't see, except for the acacias, but
Uh-huh.
and eucalyptus, which really are, completely out of their bounds now.
Right.
The other species we probably see the trees that we're, because this is not a random design, or this is a design we're going where people tell us the trees are dying, right?
Uh-huh.
So we are picking up the trees that are suffering. So what's happening is that this mortality is happening in multiple habitats of multiple species, but we are selectively going. So I don't know what percentage of trees really overall is affected, right?
Got it.
So when we look at those percentages, it will be very, very different. What this study does simply tells us that it's happening. It's happening, but it doesn't tell me how bad it is. Even if my sites have different, level of dieback from 24 to 100%, it's those sites. It's not representative of the entire populations of Northern California or the entire species. So you see, so I'm back. The way I'm looking at it, I'm just catching the worst case scenario. But what we do know, it's new. So what we know, these owners have owned this land for a long time. They've never seen this before.
So it is, even though like the different extents are different, it's still all starting at the same time.
And because yeah, but it started at the same time. It could be that all eucalyptus in the East Bay are showing symptoms, but only bays that are far away from a stream are showing symptoms, right? So again, it's happening at the same time because the conditions are changing everywhere. But the trees that are more resilient are probably in areas that are less affected by the change, and so we don't see those. So this is a very important question. I think was the perfect question for you to ask, because I wanted to make that clear, and that's why this is not doom and gloom. Because trees will still be, in some sites they will still be happy, but for how long we don't know.
Yeah, we've got, and thank you so much for that.
Thank you, Ori. The last question in particular was very important to clarify, because otherwise I know tomorrow the buzz is going to be, oh, Matteo said all of the trees are going to die.
I promise I won't say that.
No, it won't. It would, and some population will survive because they are ecologically adapted to harsh conditions, and because maybe the microclimate is good enough for them to survive even with a 2-degree increase in temperature. So that's the reality. It's just we look different. Overall, if we take an aerial image of the Bay Area, Northern California, it will be very different, and I guarantee you that a high percentage of the difference is caused by latent pathogens.
Wow.
Thank you.
So a bunch of us are working with our various cities trying to make sure that our native trees are on the street tree list. And we do have a lot of local native trees that do have long north-south ranges, coast live oak, the toyon, the buckeye, the blue elderberry. And I know that some don't, like the hazelnut. So for instance, in San Francisco, our local CNPS chapter did not ask that be put on the street tree list, because we knew it wasn't, that those are going to need a lot of hand holding. But we're really hoping that some of these other trees will have more resilience to climate change. Is there any hope for that? I just am worried about missing the insect populations if I just move up a Southern California tree here.
Well, I think the phytosanitary, that's why I think we need to start talking about this more openly, because I do know, it's as hard as, I know that we've always said local germplasm is the best, right? And in fact, we have seed maps for the conifers, right? So you can only plant a pine if it comes from the same seed map area, right? So the entire state is divided in seed maps. So which made a lot of sense, right? When climate was relatively stable. So now I really think that with as much respect as we should have for local germplasm, we really need to start a conversation. What about exploring using the same species coming from different latitude, and how to do it? Because I don't want to tell you who, but I know that some managers are already doing it, but they may not be doing it the right way, maybe necessarily. Also, the other problem, which any ecologist is aware of course, is the ecotype is very important. So you got to be very careful, because if you go and get an oak from Southern California, a coastline oak that grows, from a population near a lake or a river, the ecotype may not be any better drought tolerant than our ecotypes that are growing in a drier mountainside or hillside. So it's not as simple as just going south. And of course, ecotypes they're not necessarily genetically, it's complicated. But they're not necessarily just genetically determined. Sometimes it's plasticity. There's a lot of things that are not just genetics that determine an ecotype, which means like a plant that grows in a drier site versus a plant that grows. So we got to be careful about that. So there's another level of complexity. And I think that if we are more open about saying, you know what, I'll give you an example. Redwoods, they just don't belong here. End of story. But redwood it's native, and I like to remind people of what happened with Monterey cypress that was planted inland. They got cypress canker. But if you go to the coast between Santa Cruz and Monterey, you don't find a single tree with cypress canker. But if you go inland, you find a lot of them. And then we planted Leland cypress, which is a hybrid. And Leland cypress is 100% susceptible to cypress canker. So, and then those Leland cypress really affected the population of Monterey cypresses that were planted inland. So we have historical examples of things can go really wrong. And so I think even if a plant is native, it may not really belong to some sites, and we should start planning about replacing those plants. That we already know that you guys have that knowledge. I mean, CMPS has that knowledge, but we also have to be careful about the ecotype, and so it's a little complicated. What are we going to? I think it's pretty clear what we need to remove, if we could get everybody on board. But what we're going to put there instead is not so clear. And I'll give you an example. It's been difficult with Albany. They want to remove the eucalyptus, have become a nightmare for them, but they have the monarch butterflies, and they love eucalyptus, and the raptors love eucalyptus. And so they're stuck in this position where, and I'm not the best person to tell them, I'm not a wildlife or insect person. They're coming to me and asking me, what should we put there instead of eucalyptus? And I said, I really honestly don't know, but to serve all these different purposes, but you need to make a strong effort to remove those, because it's, and the other thing they want, the problem is a lot of people think it's cyclical and we just go back. But I think I'm starting to believe this is not just a cycle, I think my data shows it, other data is showing it, that being in a mega drought, it's not the same thing as being in a long drought. It's a completely different thing. I think it impacts the system in ways that we don't fully understand that are permanent.
Yeah.
Have I answered your question a little bit?
I think so. And Vic Paul has got a great question. Let's see if I can get Vic. There you are. Vic, you're able to talk if you'd like to.
Sure. Yeah. So you mentioned, Professor, you mentioned the, I hope I'm saying it correctly. So there's the endophytic and the other phase, which is the inside the cellular phase.
So the endophytic is the one inside the cell. Then the pathogenic is when the pathogen breaks the cells, and then the live cells. And the saprobic is when it feeds and sporulates on dead cells.
Interesting. And this is all the fungi, right?
Yeah,
So
it's the same organism. It's the same individual.
Is this phenomena reported?
Yeah.
Okay.
Yeah, that's why I show a lot of old slides, because I want at the beginning all the basic data of these organisms and how they shift lifestyle, we call it, is being discovered in the 70s and studied in the 80s and the early 90s. But it's been a very mechanistic research, so we don't fully understand how they're doing it, but we know they're doing it, and we have lots of data to show that they're doing it. And then there was a lack of interest after the initial discovery, maybe out of frustration, and maybe because for a forester like me or a woodland ecologist, which I'm kind of also, these latent pathogens they were so kind of rare, exceptional, right? That we have not been putting a lot of energy into them because it's, oh yeah, you're studying the tree that's dying because it's a redwood planted in the Central Valley. Of course it's going to die, Or oh yeah, you're studying bristle cankers on bristlecone fir in the East Bay Botanical Garden. Of course it's going to die. It's in the East Bay Botanical Garden. It should be in the Santa Lucia Mountains, So there are all of these. It became a little bit superfluous, to study them. But now, I'm not the only one. There's several people, and actually, it became really not fashionable to study them. You couldn't get funding. But now, myself and other people that are discovering, oh my God, these are the ones that are determining the range of trees, right? Because that's something that we teach. The pathogens determine the range of trees, and it's these pathogens that are doing it, right? It's these pathogens that are there to do it. So now what I hope my research does is tell people, look, we made a mistake. They were relatively exceptional because climate was relatively steady and stable. But now the climate is changing, then they're becoming major players. So yes, there is a lot of information. So it's not something that I made up. That's why when I found this fungi, immediately when I was able to ID them, I already knew that these fungi are being reported as being latent pathogens. So that really helped me significantly. Otherwise, I wouldn't have known what to do. So that's why I started to do the drought experiments and then the temperature experiments, because I already was informed by the literature that's factors that are associated with outbreaks by latent pathogens.
Thank you.
So we've got a great question about what do you do? How do land managers deal with the dead and dying trees? Is it okay to use them for wood chips and compost them? Does the temperature data kill those pathogens? What do you recommend?
Yeah, so, I made a recommendation for the East Bays and for the SFPUC. I feel a little uncomfortable using them as, the problem is the difference between compost and mulch is huge. And while I feel I haven't done the data, the research, but I've done other research on compost, I feel fairly comfortable that compost could get rid of many of them. However, some of these are very thermophilic. So again, that's why it's important to know what you're dealing with. And if you have a really thermophilic one, and if it's a major player, because obviously if it's a minor player, we're going to live with it. But if it's a major player, maybe even compost may not work. So I'm a little bit concerned. What I do know is that the faster, but okay. So what I do know, maybe I need to link the dots eventually, but I'm hesitant because I don't have a lot of data. But the faster the wood dries, the more inhospitable it becomes, and we have data to prove that. So chipping it and make yeah. So the important thing is to dry it up as fast as possible, and then maybe you can do whatever you want. Because these organisms don't produce resting spores. Some of them maybe do, but most of them don't produce resting spores that would be very tolerant of very dry conditions or anaerobic conditions and things like that. So I do think that rather than leaving the trees standing, if you chop them up, if you don't have energy, I was telling people you could just chop them up and put them in the ground, mix them with the top, which means that carbon stays inside on the side. Just don't do it next to trees, do it in areas far away from trees, because that situation attracts root rots on living trees. But just leave them on site and the soil organisms will take care of them. Solarize, or solarize them, so solarize, cut them down and solarize. So do something, whatever you can do financially. I'm not a black and white person like other pathologists. I'm very gray. Because I come from the real world. I tell people, whatever you can do that goes in the direction probably does some good. So do a cost-benefit analysis. So anything that puts them in the soil, that makes them hot, anything that reduces the volume of the of the substrate probably hurts them, because it's going to dry up too fast, or it's going to put competitors that they can't deal with these competitors.
That's very helpful. And someone else asked, there's someone who works for a Bay Area open space district, and they've been interested in best practices to prevent pathogen spread at an individual level. I mean, are there things that should we be using alcohol spray, or cleaning our shoes? What do you recommend?
That's the other, I guess in a perfect world, yes, but it's not an open space. It's not the OR, and so I think that in reality we don't really have a lot of evidence that people are really spreading them. What we do know, okay, with tools, what we do know is the wounds facilitate infection by latent pathogens. So that we know. And so management should try to reduce wounds in trees, which again, if you have a super dense woodland, it's almost impossible to do. But if you have a more landscape setting, reduce wounds, that's important. And in agriculture, they do recommend to take, when you have a branch that's dying, because dieback starts in the branches. Maybe that's something I should have explained. It's one branch at a time, and then sometimes the stem dies like a branch, basically. So what they do in agriculture, where the trees are very valuable, they take away the branches that are dying. But I don't see this. But maybe on very high value trees in an urban setting, you could do that, right? So you could do some pruning. That would potentially decrease the inoculum. But again, if it's the problem, these organisms spread in the air. So again, maybe avoid wounding would be the best thing and the most economical thing.
That's very helpful. And then Connie Barlow's got a great question. Because so many of us who are listening really care about native plants. Do you recommend assisted migration northward, upslope, or on north-facing slopes?
Do I recommend? I conceptually recommend it, but I think, as I said earlier on, it's not as simple as moving a tree from point A to point B. So, and I'm very worried about this issue of the ecotype. So I'm very worried that we may make some big mistakes, because that's going to be a big investment. But I don't see anything wrong with it, especially if it's what's happening already, right? It's what's happening in the Sierra Nevada. Things are moving up, so trees can move at a certain pace, because individuals are sessile, but obviously population move through seed and pollen. So if the climate changes too fast, we may lose entire populations, but we could save populations by moving them up ourselves. So it has to do with the rate of change. And again, different species, different, ability to be resilient or adapt. But I don't see anything wrong with us doing that. I mean, certainly moving upslope some trees is 1,000,000 times better than going to Australia and finding another eucalyptus species, which we may think is more drought tolerant. That would be the worst possible thing we could do. So let's move trees up a little bit. That's okay with me.
That's okay. That is, they've got another eucalyptus species on the San Francisco.
Yeah, I know.
It's crazy.
Yeah, because the problem is, so that's just in between us, is that in Southern California they've experimented with so many species, and I think some people are going there and maybe not getting the red gums because that's too big, too obvious, but they're picking these more obscure species of eucalyptus and testing them up north, and that's definitely something that I don't encourage at all.
Thank you. I will be quoting you. Is there any information about the soil health, the mycorrhiza fungi, anything else that could be supporting or detracting from the health of these trees, especially the native trees?
Yeah, theoretically, a healthy mycorrhizal population improves resilience. I don't know. I mean, the point is mycorrhizae are pretty ubiquitous. The only exception would be super sites, mines, or really super dry sites like the Middle East. Like I lived in Israel for a little bit, and those are situations where you gotta actually help mycorrhization, just to make sure that it's. But I mean, most of Northern California, I would say, I don't know if there's any evidence of the need to assist mycorrhization yet, but maybe things will change so much that we could because definitely it's important. So maybe some data is going to come up saying, hey, these trees are not mycorrhizal the same way they used to be. And so definitely something to look into. And something else to look into is that to look at the entire microbiome, right? Because remember, I wanted to make sure that I made it clear at the beginning, and I like to end by repeating this. These are just individual species that are part of a huge microbiome, which is essential for survival of the tree. So let's not blame these organisms. So there may be some combination of other endophytic fungi that could help the plant to resist infection, like we've shown for Myconia calvescens. We were able to show it experimentally that if they had the right combination of endophytes, they could protect it from infection by a fungus. So I could totally see, besides the mycorrhizae, that the microbiome has a huge potential. I mean, the other thing that's that I'm trying to wrap my head around is that some of these latent pathogens that are major players, they were first reported in agriculture in California. Now, I don't know, I don't want to talk cause effect here, right? I mean, and we need agriculture. So I'm not, I don't think it's, but all I'm saying is some of our native plants may have been infected by some of these. That's why I'm saying let's eliminate inoculums, but by plants that were, in agricultural settings, which would favor infection by these organisms, because it's monocultures usually, and they're grown relatively close to one another. So it's kind of the perfect setting. And then they express themselves when you're not watering them right, or whether the year's too harsh, right? So they're familiar with some of these organisms in agriculture. And I'm just working now for BLM on this is huge mortality in Nevada, Arizona, in California, of scrubby mesquite, which is a desert adapted plant. And we're actually finding two culprits that I think are driven by climate change. And I can find them in the literature on orchard trees. One of them already in California, other ones in other Mediterranean climate countries. Doesn't mean cause effect. I don't know. Maybe it's just, a coincidence. But it's maybe because people have looked into agricultural systems more closely, right? Because the livelihood of people depends directly, immediately. So there's more research in those systems. So that's maybe the only reason why. But so I'm trying to wrap my head around that. Is there a connection? But we're finding two fungi that are fairly common in mesquite, that it's causing this dieback that I think eventually ends up killing them.
That's terrifying. Mesquite are really tough plants, nitrogen fixers. They're
absolutely. And and the interesting thing is that it's honey mesquite is not affected, because we worked in we have about 8 study sites, and there's honey mesquite. Honey mesquite is doing pretty well. I mean, occasionally you have one that's dead, but for different reasons. And the ecologists have explained to me that one characteristic of scrubby mesquite is that they call it's a desert riparian species. There's no rivers in the desert, but there's underwater rivers, right? I mean, and they grow in these areas. Sometimes you can see that. You can see them following, just dry beds. And so it makes sense. while honey mesquite grows everywhere, independent of water availability. So it makes sense that scrubby mesquite, again, we have a situation where we have with two very similar trees can grow in the same environment, but there is a difference. That is how close you grow to water availability. And those are the trees that are suffering the most, probably because of some changes in the temperatures in the desert or water, or something. But I think it's environmental from what, it's strongly environmentally driven.
Wow.
And then the pathogen comes in and kills the tree. So yeah, so it's not just these dots here, it's happening. This is just what we did, starting from the acacias. And these are basically all the answers of all the calls I got from different ownerships. So that's what I'm showing. So if people were not calling me, they're not part of the study.
This is very sobering information. Thank you so much for sharing it with us. And thank you everyone for all the wonderful questions.
Okay.
Any last comment?
Are we done? Is there one more burning question or?
No, I think we're done.
Okay, well, thank you so much. And again, I'm not saying that all the trees are going to die, okay? But it is one mechanism that's going to take out parts of the populations of our trees now.
Thank you very much for this
Thank you very much for inviting me, and invite me again to talk about something else, maybe less dramatic. But as a pathologist, it's hard. The talks are always, people want to hear about, trees dying. And
it's, yeah, the world we live in. Thank you so much for all this good information.
Well, you're welcome, and thanks again for inviting me. Bye-bye.
Good night.